Hi everyone, we're looking at a process known as mitosis in this chapter. And mitosis is a way that a lot of different cells divide. Specifically, these would be eukaryotic cells. Prokaryotes like bacteria and archaea, they're going to divide a little bit different way, which we'll actually mention at the end of this chapter. And then also there's a select few cells called the gametes, so sperm and egg, which are going to divide a little bit differently than what we talk about here. But by and large, most of the cells in eukaryotic organisms will divide through this process known as mitosis. So before we can talk about cell division, we should talk a little bit about the genome. One of the main goals of one cell becoming two new cells is to make sure that the two new cells that are formed are a perfect copy of the original cell. So you start off with one cell, and then it gets copied into two new cells. These two new cells have to be identical clones of the first. And part of the way that this happens is to make sure that all of the DNA that's inside this cell gets copied and then packaged in exact proportions to the following new cells. So the genome refers to all of the basically DNA, or all of the what are called genes. And a gene is just basically a region of DNA that codes for some sort of trait. So we're made up of all sorts of traits. All organisms have lots of different traits. Could be the texture of their skin, the color for you and I, the color of our hair and our eyes, or the shape of our nose. All of these different features of the organism are going to be coded by different parts of DNA. So one part of DNA might code hair color, another part of DNA might code eye color, and so on and so forth. Well, those specific regions are called genes, and if you take all of the genes out there and you organize them up, it's the organism's genome, the collection of all of the genes. This is made up out of DNA, and recall that DNA has a double helix, meaning there's basically two kind of strands of two sides of a ladder, think of it, and then in between the sides of the ladder are the rungs. And the rungs are where you have complementary base pairs. And remember that C always pairs with G, and A always pairs with T when we're talking about DNA. Well, if you take that double-stranded helix, kind of like you're seeing here, and you wrap it around this structure, which is trying to illustrate what's known as a histone protein, then you have basically a little DNA wrapped around a ball of protein. This is called a nucleosome. And again, it's just a ball of protein that has DNA wrapped around it. And then this allows the DNA to kind of coil, and then actually all of these nucleosomes, kind of these more organized masses here, can even condense on one another to start to form something like this, where you have a whole bunch of, you got one DNA that's wound around a histone, and then right after it leaves, it winds around another one, and then it winds around another one, and then it comes and winds around another one. And then before too long, you've got all of this DNA wound up and getting scrunched together as the nucleosome strands are themselves coiling and condensing. So this is when we start to form this stuff called chromatin. Chromatin is basically just the molecule DNA and then all of the associated proteins involved to pack it down and fold it down. That is the purpose of these proteins, is to just pack it down, but and also give it a little bit of structural integrity, so it's not just like a bunch of spaghetti that's going to break apart. Okay? Now, when you have a nucleosome compacted fiber kind of like this, you can actually take this and then form a whole bunch of loops. So this whole strand that you're seeing here, that's just a small region of what would be maybe a loop that looks like this. And then there could be another loop, and then there'd be another loop, and another loop, and another loop. And now you've got these DNA, these histone-wrapped nucleosome, or the DNA-wrapped nucleosomes, all packed in on one another, and they are in turn folding into this huge megastructure called the chromosome. Now, keeping in mind, a chromosome has got tons and tons and tons and tons and tons of DNA wrapped around and around and around and around and around and around. If you were to like shrink yourself to like this size, here you are walking around. I mean, you could peruse through all of this DNA for years and years and years, and you still wouldn't get to one end of the chromosome to the other. So it is an incredible amount of DNA packed all within one tiny chromosome, and you and I have a total of 46 chromosomes. So that is a lot of DNA. And I'm always astounded when I think about this. How does the cell find anything in there? You know, I always make the joke, I can hardly find the proper shirt that I want to wear in the morning. How can a cell go into a molecule of DNA that's got like 1,000 genes on it, tons and tons of information, and code one of them for some specific protein that needs to be made? It's just truly astounding. So this is the nature of chromosomes. Now I bring this up because mitosis is really one of the main goals, is to get chromosomes duplicated and then to send one this way and send one this way and make two new cells with identical chromosomes. We already talked briefly about what genes are again, but just recall that genes are going to be a certain segment of DNA that codes for some sort of trait. Right, and these can be like thousands and thousands and thousands of base pairs long, each gene, or it could just be a couple hundred base pairs long, maybe. Depends on how complex the trait is, or how complex the structure is that is being coded for by the DNA. Now, when we're talking about genes and genetics and things like that, there's a couple important terms I'll throw up here. Alleles. What these are is this is variations of a trait. So, for example, a trait might be, let's go with just this standard example here, eye color. All right, that would be the trait, eye color. But you, in that trait, could have different alleles of the trait. So, like allele number 1 might be blue eye color. Oops. Allele number 2 might be brown eye color. So, and so on and so forth. You could have green and hazel and even red. So those are all different types of alleles of the trait. So when you see that word, that's what that means. And that will play more of an important role when we get to talking about meiosis in the next chapter. Another word is this one called phenotype. Phenotype is the actual appearance of the trait. Whereas the genotype is a little bit different. This is more like the code for the trait. So, for example, on some part of DNA, let's say between right in here and right in here, let's say all of these little base pairs, they store the information required to build some sort of protein that give my eyes a blue color. Okay, so my genotype would refer specifically to the genes, so specifically to the A's, the C's, the T's, and the G's, the sequence of all of these bases here. Whatever that sequence of that is, that would be the genotype. Now, once this information actually gets used to build something, in our example, the pigment that makes my eye blue, a blue color, then the actual appearance of that is the phenotype. Okay, so I could have a bunch of base pairs here coding for blue eye color, and that we would say is the genotype, whereas the phenotype would be the actual appearance of the trait, meaning my blue eyes. So these can be sometimes easily confused. And just think about it this way. You could think about the genotype as being like the blueprints to build something, and the phenotype is the actual building that's built from the blueprints. Okay. Here's kind of a cool picture which shows what's known as a karyotype. So a karyotype is kind of an assessment of an organism's chromosomes. All the chromosomes get stained and then kind of photoshopped, so to speak, and arranged and categorized. And what you're seeing here is this represents what a nucleus might look like prior to cell division. Generally, the chromosomes aren't really super condensed. In this picture I showed all of this stuff super condensed into this nice discrete packaged bundle of DNA called a chromosome. Well, in reality, they're only really like that before the cell divides. In the normal day-to-day life of a cell, they're kind of uncoiled a little bit, so it looks a little bit more like a bowl of spaghetti, kind of. Well, right prior to division, all of those chromosomes are going to condense into nice, discrete packets, which look like this. Okay, so we call these things chromosomes. Now, you and I have what are known as 46 chromosomes, or we have 46 chromosomes, and there's a whole bunch of different genes and all assorted through all of these different chromosomes. Now, what's interesting about you and I is that we actually have 23 pairs of chromosomes. Is kind of a good way to think about it. So if you arrange all of these chromosomes that might be found in a human, let's say, into this nice karyotype, you see that there are 2 chromosome number 1s, there are 2 chromosome number 2s, there are 2 chromosome number 3s, there are 2 chromosome number 4s, and they are numbered by the relative shape. You know, you can see chromosome 1 is the longest one, and it's got kind of an attachment point right here in the middle. Chromosome 2 is a little bit more, is a little bit less tall, and then you've got like even really, really small ones like chromosome 20 or 21, 22. So they kind of arrange them by shape. Now, this gets us to an interesting an interesting difference between something that's diploid and something that's haploid. In this example here, this is actually showing something that's diploid. Diploid means that there are 2 sets of chromosomes. Haploid means there's only 1 set of chromosomes. So the reason, and you and I are diploid, and so are all the other animals and plants out there. The reason why there are 2 sets of chromosomes is because you inherit 1 set from your dad, and you inherit 1 set from your mom, and so your dad contributed 23 chromosomes, your mom contributed 23 chromosomes, and that makes up 2 sets for a total of 46 chromosomes. Well, what we call these things are homologous chromosomes or homologous pairs. So chromosome number 1, this is maybe, let's say, the one that came from dad, and the other one is one that came from mom. Now, these 2 homologous chromosomes are not identical because, you know, your dad's DNA is not going to be identical to your mom's DNA. If it were, they would, you know, your mom and dad would actually look completely identical. But I'm guessing there are some pretty significant differences in the appearance of your mom and your dad. So if that is the case, that they do have, you know, their own sets of DNA. So even though these are the same shape, these chromosomes, and they also contain all of the same genes, the actual DNA itself is going to be different. So for example, like right here, you might see that there's or you might find a gene for blonde hair. And let's say that whatever it is that makes your hair a certain color, that gene is found on chromosome number 1. Whether that's true or not, I don't know. But let's just, for the sake of argument, say it is. Well, let's say maybe your dad had blonde hair, and so he passed you this chromosome with the blonde hair gene on it. And then at that same spot on your mom, the chromosome your mom gave you, let's say maybe that gene codes for brown hair. So you can see that, you know, at about the same, what's called the locus, or the loci, the same like region of the chromosome, you're going to find the same genes. But each gene is going to be probably a little bit different on either chromosome. So that can be a little bit tricky to understand, is the difference between, you know, what a homologous chromosome is. So basically, we all have 22 pairs of chromosomes, and then 1 pair of what are called sex chromosomes. So this particular one is an XX, which would be a female. If this was a male, it would be X and a really small one called a Y. Almost all of our cells are going to have 46 chromosomes like this. They're all going to be diploid. Now, the only cells, for you and I at least, that do not have this diploid nature are going to be what are known as the gametes. And the gametes are things like sperm and egg. Rather than being diploid, rather than having 2 sets of chromosomes, sperm and egg only have 1 set of chromosomes. So they only have 23 chromosomes. They have 22 normal chromosomes and then just last sex chromosome to make 23 total. Well, when the sperm meets the egg, well, the 23 and the 23 combine to form this type of diploid situation that you see right here. Okay, so that's a little bit about chromosomes, genetics. We'll talk a little bit more about how that all comes into play here in the next chapter, but do be aware of that term, homologous chromosomes, because that's going to be an important thing to keep straight. All right, your first review question then is right here. Go ahead and pause the video, work through these, and see what you come up with. I'll go through them in just a minute. All right, here we are. Part A, skin cells, are they haploid or diploid? The answer here would be diploid. All cells except sperm and egg are going to be diploid. So that leads us to the next question. Egg and sperm are haploid. Describe the basic DNA molecule structure and the double helix. Basically, there's going to be two sides, and in between the two sides there are nitrogenous bases that are complementary paired with one another. And remember that A always goes with a T, and C always goes with a G, and G always goes with a C, and T always goes with an A. And then if you were to spiral this thing, wind it up, that would be the shape of the double helix. Now the structure of one chromosome, basically DNA gets wound around a histone protein, and that forms a structure called the nucleosome. A whole bunch of nucleosomes compact, and that forms a chromosome. What is a pair of homologous chromosomes, and are they identical? The answer here, a pair of homologous chromosomes refers to chromosomes of the same that bear the same genes and the same shape as one another, but they come from different parents. So one would come from mom, one would come from dad. Are they identical? Kind of. They have the same. They bear the same genes, but because those genes are different, the DNA sequences are going to be different. So they are not technically identical. How many individual chromosomes are in a human body cell? That would be 46. How many pairs are, so how many individual ones? Okay. Then this asks how many homologous pairs are in a human body cell. That would be 23 homologous pairs. All right, moving on to the cell cycle. This is a figure which shows the cell cycle, and a cell spends most of its time in what's known as interphase. Interphase is kind of like the daily normal life of a cell while it's either hanging out or preparing to divide, but not actually undergoing the dividing process. And it's kind of interesting. Sometimes cells can be in this state which is known as the G subset 0 phase. And this is basically just kind of the normal life of a cell. They're just kind of going around doing their own thing. They're not thinking about dividing, they're not preparing to divide at all. They're just kind of cranking out energy, doing all this fun stuff that cells do. And they do a lot of stuff. I couldn't even tell you all the stuff that a cell does, but they do all sorts of stuff. Okay, now when a cell decides that it's time to divide for a variety of reasons, it enters what's known as the G1 phase of interphase. Okay, now G1, so it kind of like comes out of this normal life and now enters kind of the preparations to divide. G1 is characterized by the growth of the cell. Basically, it's accumulating. A couple of things that happen in G1 is that it's accumulating building blocks. So these are going to be things like amino acids, lipids, maybe some nucleotides, all of the different kind of atoms that it needs to build stuff. It's also going to be accumulating energy. And overall, the cell is just kind of getting bigger. because it's preparing to divide. Then what happens is we enter what's known as the S phase of interphase. And this is the point at which the cell is going to start copying all of its DNA. So it's going to be making lots and lots of DNA copies. And you can imagine this is quite a feat, right? Because there's tons and tons of DNA in a cell, and to actually go through and copy every last little nucleotide down to the T, make perfect copies of every single chromosome, and mind you, there are 46 chromosomes. That's a lot of work, and that takes a lot of ATP to build all of that, a lot of resources and a lot of ATP to build all of those really long DNA molecules. So that's kind of why the cell enters G1 first to accumulate all of those things, and then it's allowed to progress onto the S phase. Then what happens in the S phase is kind of shown here. So here's a pair of homologous chromosomes. Okay, so again, keep in mind one of these came from dad and one of these came from mom, and they have the same genes on them. Well, there's this region on a chromosome called a centromere. And a centromere is going to be the place at which a duplicated chromosome is going to kind of be built and attached to the original chromosome. So after the chromosomes go through the S phase, they will have replicated, meaning they'll look something like this. They'll have what are called sister chromatids. And what a sister chromatid is, this is an identical copy of the original chromosome. Okay, so this is where something can get a little confusing. Homologous chromosomes are not identical. One came from mom, one came from dad. They have the same genes, but different different alleles on those genes. So there's going to be different DNA sequences. Sister chromatids, however, these are exact copies of the original chromosome. So sister chromatids are identical. And right after the S phase, every chromosome is going to have a duplicated, what's called sister chromatid, and they are attached together at this point, which is called the centromere. Okay, so that's what the story looks like inside the nucleus after the S phase. Now, a lot of ATP was spent here, and a lot of resources were consumed here to try and build all of those really extensive DNA molecules. So then the last phase before it gets ready to divide is the G2 phase, and this is just to kind of recuperate all of the expenditure of energy. So things that happen in the G2 phase, there's going to be more accumulation of energy to replenish all that was lost. And then another thing that happens is that some of the organelles actually in this are going to kind of be dismantled and duplicated. A lot of the membranous organelles, like the endoplasmic reticulum and the Golgi apparatus, they kind of start fragmenting and then kind of going their separate ways to form two new organelles. Now, once all this has happened, all of the organelles have been kind of duplicated and all of the DNA has been duplicated, then the cell is ready to divide. So it enters out of, it leaves what's known as the interphase of the cell life, and it enters the mitotic phase. And this is the actual process of the cell going through its division. And it's formed of mitosis, which is kind of the process of going through division, and then cytokinesis, which is the final last kind of separation of the two cells. Right, here is another review question for you. Go ahead and pause the video and work through these. On these ones, I didn't really talk about some of these, so I'll kind of answer those for you as we proceed. All right, part A, the two main phases of the eukaryotic cell cycle. We're going to have interphase, and then also the mitotic phase. What are the three stages of interphase? Well, there's G1, there is the S phase, there is the G2 phase, and then there's also kind of this weird one, G0, where the cell's just kind of doing its normal thing. G1, we have the accumulation of building blocks, the accumulation of energy. In the S phase, we have the duplication of all of the DNA, so a lot of DNA is made or synthesized. That's going to use up a lot of those resources that we gathered in G1, and now we're going to need to replenish that energy that was expended during that process. So in G2, we're going to duplicate the organelles and also replenish all of the energy that was lost in the S phase. So keep those kind of in mind. Could you kind of differentiate which one is which? If I gave you a little prompt and told you a little detail, could you tell me if it was, G1, S, or G2? Okay, then the functions and the purpose of the mitosis. This is going to be to make 2 new cells from 1. And these are going to be, what's important here is that these are identical cells. What type of cell division produces 2 new cells with identical diploid DNA? That's going to be this process that we said is mitosis. Which type of cell division produces cells that are haploid? That's going to be this one called meiosis, and we'll look more at this later, so don't worry too much about it now. What type of cell division is asexual reproduction? That one is a little bit beyond what I want to talk about here, so that kind of stuck in here. Cell division precedes sexual reproduction. That would be mitosis. How many sister chromatids would be present in one of your skin cells after the S phase? All right, so keep in mind we're not really too concerned about these. Okay, sister chromatids present in one of your skin cells. So a skin cell, is it haploid or diploid? The answer is that it's diploid, because all cells except gametes are diploid. So if it's diploid, it means it has 2 sets of chromosomes, so it's got a total of 46 chromosomes. And then if each one of those chromosomes duplicates into 2 sister chromatids, much like we saw in this figure here, where you have 1 chromosome duplicating, forming 2 sister chromatids. If that was the case, then 46 chromosomes would then turn into 92 sister chromatids. All right. Okay, so now the actual process of mitosis. Mitosis is divided into these stages. The first is called prophase. In prophase, there's a couple of things happening. Basically, one of the biggest things is that the nuclear envelope is kind of starting to break apart, so that's why you see this kind of dotted line starting to form here. It's trying to show you the nuclear envelope is disintegrating. Also, what's happening is the formation of what's known as the mitotic spindle apparatus. Mitotic spindle apparatus. And that's these little blue poles that you see kind of coming out here. They're coming out of this interesting organelle, which is called the centrosome. Now, when you see centrosome, make sure you remember to yourself that this is not the centromere. The centromere is the place that 2 sister chromatids will attach to one another. The centrosome is comprised of 2 centrioles. It's an organelle that makes these little blue pole thingy-mabobbers that are coming out. Okay, and those blue pole thingy-mabobbers are going to be really important for the cell division process. So that's kind of what's going on in prophase. There's a little summary here you can read about it. You have spindle fibers emerging from the centrosomes, nuclear envelope breaks down, nucleolus disappears. Oh, that's kind of interesting. But remember, the nucleolus is the place that ribosomes are made, deep down into the nucleus, so that kind of disintegrates. Oh, one thing I forgot too is that the chromosomes are becoming pretty condensed. I said earlier that chromosomes are a little bit looser prior to dividing, and then once they decide to divide, they get kind of condensed more, like that picture we were seeing earlier. So that's what's going on in prophase. Prometaphase is all about this structure known as the kinetochore. And the kinetochore is basically a little region on the chromosome that forms. It's kind of like a little sticky pad that the spindles can attach to. So you can kind of think about it as being like a little Velcro pad. And these little mitotic spindles, which are coming from the centrosome, they go and they land on that thing and they attach to it. And that's going to be a really important setup. So you're going to see that the kinetochores appear, and then the mitotic spindle is attached to the kinetochores. So you have these things sticking on to these little kinetochores. Before anything can really happen, all the chromosomes have to have one of those spindle fibers attaching to their kinetochore. And then in metaphase, basically all of the chromosomes are lined up right in the middle in this region, which is called the metaphase plate. And here, metaphase plate. And then as soon as they're all lined up and they're all attached, then the very next part is the anaphase, and that's going to be where the sister chromatids are actually kind of pulled apart from one another. And so what's happening is that these microtubules are actually kind of like disintegrating, and that's going to be pulling the chromosomes back towards the centrosome. And that's important because it pulls all the DNA over here, and it pulls all the DNA over here, and then you've got basically kind of two new cells which are still in the same cell, technically speaking, but they're all the new DNA is over here and all the new DNA is over here. So you're separating out all the cellular contents into what is called two different poles of the cell. So this would be kind of one pole of the cell. Here is another pole of the cell. The last is telophase. The big event that's happening here is the reformation of the nuclear envelope, as you see here. And at this point, all of the chromosomes have arrived to their separate sides of the cell. And all of the mitotic spindles can then, at this point, break down because they've sort of served their job of pulling those sister chromatids apart. The last thing that needs to happen then is what's called cytokinesis. And this is basically the final kind of separation of the cell into two new cells. And this is going to happen differently depending on if we're talking about an animal cell or a plant cell. So here's a good picture that kind of shows the difference. Here's showing a cell that all of the cellular contents have been duplicated and now have made it into the two separate poles of the cell. Well, what happens is that there's going to be a bunch of actin filaments that wrap around here, and they're going to kind of cinch down and actually kind of just basically pinch off that region. It forms what's called a cleavage furrow. This little indentation here as it's getting pinched in. It's called the cleavage furrow, and then eventually yields two new cells. Whereas here it's a little bit different. You know, an animal cell has got a plasma membrane, so it's as kind of the outer boundary, so it's kind of a little bit more flexible. Plant cells have a more rigid cell wall, so it's not as easy to just kind of pinch them together and create two new things. So it's a little bit, how this works is a little bit different in a plant cell. Rather than actin filaments pinching the middle, what happens is that little pieces here are actually constructed. And almost like you can think about it as almost being like a brick wall is produced between the two poles of the cell. And then eventually that brick wall is kind of more solidified into forming a cell plate. And now there's two separate cells from what was just one cell. So animal cells, there's a cleavage furrow, and that's because there's a plasma membrane. Plant cells, there's a cell wall, and so it forms not a cleavage furrow, but it forms this thing called a cell plate, which is like a wall. Alright, here's review question number 3. Name the chromosomal structure to which mitotic spindle microtubules attach, and then list and summarize the steps of mitosis. Go ahead and pause the video, and I'll work through these, and I'll go through it in just a minute. Okay, the chromosomal structure is going to be this thing that's called the kinetochore. The processes of mitosis look something like this. There's prophase. There is prometaphase. There is metaphase. Anaphase. Telophase. And then lastly, cytokinesis. And then what's happening in each of these? Prophase, basically the nuclear envelope is breaking down, the mitotic spindle is forming. Also, another thing I forgot to mention prophase is a lot of this, the cytoskeleton is being dismantled. You know, all of those microtubules and actin filaments which give the cell its shape. well, they're kind of being broken down to make room for all of this movement of stuff that's going to be going on. In prometaphase, the big thing that's happening is the kinetochore is formed and the microtubules attached to it. Also, in these two, the DNA is condensing into more discrete chromosome units. In metaphase, those chromosomes are fully condensed and they're lined up at the middle, and now they're going to be pulled apart. And then as they're being pulled apart, we'll call that anaphase. Once they've been mostly pulled apart and they've arrived at two separate poles of the cell, and the nucleus reforms around those chromosomes, then we're into telophase. And then the last thing is for the cell to actually do its final division and actually form two new cells from the one cell. Alright, well, that's a little bit about the process, and now we're going to talk a little bit about how all of this is controlled or governed. And this is going to be really important because, especially relevant to our society, because cancers, a variety of cancers out there, most of them have their kind of origins in something that goes wrong with the controls of the cell cycle. A cancer cell is one that divides uncontrollably. It just divides, and it can't stop dividing. And so usually those are formed when whatever mechanism is put in place to normally keep the cell cycle monitored, that mechanism fails. And when that mechanism fails, it can lead to a cell that can not stop dividing, which is known as a cancer cell. So this is pretty important stuff to talk about, and there's a lot of research that's been done on the cell cycle for that reason of the devastating effects of cancer. Now, how the cell cycle is controlled, it can either be external events or internal events. External events would be things like the death of a nearby cell, or even a variety of different hormones acting on a cell. Some of these are some examples of things that can happen outside of the cell that can work to trigger or encourage a cell to divide or not divide. You know, the hormone that's listed here, growth hormone, that's one that's released a lot during puberty, and that's going to be responsible for making a lot of your bones grow at a faster rate, and your muscles grow at a faster rate, and all that stuff. So that's kind of an external stimulation there. But the cell has its own kind of internal, what are called checkpoints. And there's basically 3 checkpoints in this whole cell cycle. Okay, so there's one that happens right after the G1 phase. There's one that happens after the G2 phase, and then there's one that happens at the M phase. And each one of these has kind of at each one of these checkpoints, there's a series of criteria that need to be met in order for the cell cycle to progress. And if those criteria are not met, then the cell cycle will stop, and the cell will actually be undergoing a process known as apoptosis, which is basically the programmed death of the cell. So the type of things that the cell is looking for when it's at the G1 checkpoint. So the cell has just accumulated a whole bunch of building blocks. It's broken down all of its It's just accumulated all of its building blocks, and it's accumulated a lot of its energy stores. And then before it gets ready to copy all the DNA, there's going to be a couple of questions the cell asks itself. Now, keep in mind, cells don't actually ask themselves questions, but you can, if it's helpful, you can think of them as asking these questions. First, they're wondering if the cell is the appropriate size to divide. Am I large enough for this division event? Do I really want to commit to this event? So they're going to kind of assess the size of the cell. If the cell size is appropriate, it's big enough for a division event, then we'll check that off the list. Another thing that the cell might wonder is, are there adequate reserves? You know, in this whole process, there is supposed to be enough DNA and enough ATP and enough building blocks to proceed, and if there aren't, then that's trouble. So it's going to make sure it's got enough supplies on hand. And then the last thing is that it's going to make sure there's no DNA damage. And this is pretty cool when you think about there being like molecules that go through and read all of the DNA and see if there's even one little small, minute, tiny little break or incorrect base pair. And I mean, we're talking about millions of base pairs that these proteins that don't have any sort of eyes or ears or brain, they're going through and they're reading all of this code. and making sure that there's no damage in all the millions and millions of base pairs that you have in your DNA. Man, that's extraordinary, isn't it? Well, if there is no DNA damage well, let's hold on a second. If there is DNA damage, there's a couple of things that can be done. The damage can actually be tried, the cell can try and fix it. And if it does fix it, and so there's no more DNA damage, then the cell will proceed to the S phase, and will start copying all of its DNA. And this is particularly important here because the cell doesn't want to copy a bunch of DNA that's damaged. Otherwise, there's a high risk that weird mutations are going to happen that lead to cancer cells. So it's generally not good for a cell to have kind of messed up DNA and replace itself and replicate itself and create clones of itself with that broken DNA. So it's going to try and do whatever it can to fix the DNA before it replicates. So then after the S phase, then we enter the G checkpoint, and basically the chromosomes the cell is wondering if all of the chromosomes were successfully duplicated. Okay, and if they were, great, we'll proceed to the mitotic phase, and then in that is the M checkpoint, what's called. Basically, the big criteria here is to make sure that the kinetochore of sister chromatids, each and every single kinetochore is attached to a microtubule. You can imagine if, let's say we're in the middle of metaphase of mitosis, and all of the chromosomes are lined up in the middle at the metaphase plate. Well, in metaphase, or then in anaphase, all of these mitotic spindles are here, and they're going to be pulling the sister chromatids to the prospective poles of the cell. Well, if there's a chromosome, let's say right here, that didn't have a spindle attached to it, then what would happen is that all of these would get pulled over here, and all of these, and this one would get pulled over here, and then this one right here that doesn't have anything attached to it, it would just kind of stay in the middle. So there'd be one chromosome here, there'd be a bunch of chromosomes there, there'd be a bunch of chromosomes here, and the one that didn't get attached would be stuck in the middle. And whether it goes over here or whether it goes over here when the cells divide, it's kind of up in the air. If it did go over here, then that would mean that when the cell divides, this one is one short a chromosome, and this one is one extra of a chromosome. So that's a pretty important thing to make sure that all of the sister chromatids are attached to the kinetochore, and that's going to be one of the things that the cell checks for to make sure that's the case before it completes this whole mitotic phase. And if there's any one of these checkpoints that the criteria are not met, boom, the cell is turned off. It undergoes programmed death, known as apoptosis. The organism would much rather destroy one of its own cells than risk raising an army of cells that have something that's not quite right about them, meaning the DNA is damaged in some way, and it's going to maybe lead to a whole variety of mutations entering into the organism. Alright, so some of the things that actually do this monitoring that we've been talking about, that actually control the checkpoints, you can break these into two. There's probably a whole bunch of different molecules out there. We're only going to talk about a few of them, and you can break them into two categories. There's the positive regulatory molecules. These are going to be the ones that want to encourage the cell cycle. They're going to try and make the cell cycle progress or can keep going forward. And there's two here, the cyclins, and then what are known as the CDKs. And you can see that basically if you were to measure the amount of cyclins, you can see that they kind of increase right in between these phases. Now you don't actually have to know all the different cyclins here, E, D, A, and B, but I'm just trying to show you that, let's say, when a cell is getting ready to go from G1 to S, one of the triggers or one of the things that promotes that is this accumulation of cyclin E. And then once it reaches a certain point, it's going to say, okay, yep, go ahead to the S phase. And then likewise, cyclin A, after the end of the S phase, when cyclin A is accumulated enough, it's going to go, okay, yep, go ahead and go on, proceed to the G2 phase. So it's the amount of these cyclins that are building up inside the cell which can kind of regulate what's going on here. But which cyclin is happening where, you don't need to know any of that, so you don't really need to know this graph. Cyclins usually work hand in hand with these molecules called CDKs. So how it works is that a cyclin is going to attach to a CDK. When that happens, it actually recruits an enzyme to put a phosphate group on the complex. So now we've got a cyclin, a CDK, and this enzyme has come along and attached now a phosphate group. Well, then this CDK, what we would call a phosphorylated CDK cyclin complex, is now going to go transfer its phosphate onto some other target protein, which is then going to go do a bunch of stuff like tell the cell to turn on and proceed to the next phase, whether it be the S phase or the G2 phase or whatnot. So that's a little bit about how that works. Again, you don't really have to know all of the specifics here, but just know that CDKs are molecules that associate with cyclin to help turn on the cell cycle. All right, then we also have the negative regulatory molecules. These are ones that are going to tell the cell cycle to stop. They're going to try and inhibit the cell cycle when it needs to be inhibited. And a couple of interesting examples here. We have one called retinoblastoma protein, or RB, and then these ones are big ones here, P53 and P21. They're named P53 and P21. It's kind of an uncreative name. It's just a name that kind of indicates how big the protein is. So P53 is going to be one size, and P21 is going to be a different size. Well, in either case, these are going to function most at the G1 checkpoint. And these are the things that are kind of detecting if there's any DNA damage. So earlier when I said, back here, when we've got cells that are looking to see if there is DNA damage, the molecules that are actually going out and perusing the DNA and responsible for this whole part is going to be mostly these. Yeah, that RB, P53, and P21. Okay, those are the ones that are doing this. And if damage is found, what they can do is they can go try and find some repair enzymes. They're going to go try and recruit some enzymes that can slice out the bad part and put in a new good part and keep that DNA nice and clean and functioning. If the enzymes are recruited to repair the DNA, but the DNA is just beyond repair, it's too broken, it's too messed up, then those molecules, P53, P21, and RB, they're going to collectively work together to tell the cell that it needs to just shut down, close up shop, and be destroyed. So that is what we call apoptosis. All right, so a little bit more here about how cancer fits into all of this. The genes that are going to be coding for positive cell cycle regulators, so again, these are the cyclins, at least for in our class, we just know about these ones here, the cyclins and the CDKs. The regions on the DNA that code for these molecules, these regulatory molecules, those are a collection of genes called the proto-oncogenes. What can happen is that a normal proto-oncogene, which codes for these things, it can get kind of messed up somehow, whether it be sunlight hitting the DNA and or just some random chemical that really put some sort of mutation in that gene, so that if the gene, the proto-oncogene, is mutated, then it's no longer probably producing cyclins and CDKs that work as they ought to. And so this actually then is going to start producing something else that could be bad. Okay, so if a proto-oncogene is damaged, it no longer can produce the cyclins and CDKs, it starts producing something else which could be bad. And when this proto-oncogene is mutated to something, some other gene that produces this whatever substance, we call that a oncogene. So proto-oncogenes get mutated into what are called oncogenes, which are called cancer-forming genes, because they're going to basically be producing, they're going to be producing regulatory molecules which don't work as they should, and that can be bad. Then there's also the tumor suppressor genes. These are kind of the flip-flop. These are genes which code for the the negative regulatory molecules. So the examples here again were PB and then P53 and P21. So the regions of DNA that code and the information required to build these regulatory molecules, those are called the tumor suppressor genes. Now, if those get mutated, then that can also lead to bad news, because then these things, which are normally supposed to inhibit the cell cycle, if they've been mutated, then they may not work to inhibit the cell cycle when they need to. So here's kind of a picture that shows this. Let's say there's a normal p53 gene. Well, a normal p53 gene, if there is any kind of DNA damage or there's any sort of weird stuff going on with the cell cycle, the p53 gene will code for a p53 protein, and that p53 protein, assuming there's no mutations in the gene, is going to be pretty healthy, and so it's going to do its job. And its job is to stop the cell cycle, figure out what's wrong, and try and repair the DNA if it can. If it can't, then its job is to just destroy that cell. Okay. Now, let's say then we have a mutated p53 gene. So whatever region of DNA that codes for p53 was either hit by too much sunlight, or there's some sort of weird chemical thing, or just random chance that is causing that mutation. Well, then, even under the same circumstances as the normal one, it could be the DNA damage or just any whatever abnormality or hypoxia or whatnot, what can happen is that the p53, the mutated gene, is going to code for a mutated protein. Okay, so the mutated gene codes for the mutated protein, and then this doesn't. Oh. Excuse me, I must be tired. Then the p53 gene then doesn't do what it should, because it's got this big chip off of it. It's broken. So rather than stop the cell cycle and repair DNA damage or lead to apoptosis, the cell cycle continues. And now you've got a cell that's maybe has some DNA damage or something weird going on, that's reproducing itself uncontrollably. The brakes aren't being put on it. It's just reproducing. So that's kind of the definition of what a cancer cell is and how this all works out. And nearly all forms of cancer, when they kind of look at them, they usually find that there somewhere along the formation of the cancer there was a mutated p53 gene. So it's kind of a big region for interest there in terms of cancer research. All right, one last question here, and then we got one last thing to talk about, and then we'll call it a day. The purpose of the 3 checkpoints during the cell cycle, this is just to basically make sure that a cell doesn't divide when it shouldn't. What is a cancer cell? How are they formed? A cancer cell is a cell that has uncontrollable growth. It just divides and divides and divides. It reproduces uncontrollably. And the way that they're formed is by some sort of irregularity in the control mechanisms. So probably some sort of mutation in whatever gene that codes for a protein that is supposed to turn on or turn off the cell cycle when it needs to be. The proto-oncogenes and tumor suppressor genes. Proto-oncogenes, these are ones that code for positive regulators, so the ones that are going to encourage the cell cycle. These are the genes that code for the negative regulators, the ones that are going to try and halt or stop the cell cycle. And then explain how a p53 mutation and p53 tumor suppressor gene potentially lead to formation of cancer cell. Well, if there's a mutation in the p53 gene, then it's likely that there might be a mutated p53, and the job that p53 normally does is to stop the cells from dividing if they've got some weird DNA thing going on. Well, if the mutated p53 is unable to do that, then that mutated p53 gene is going to lead to the formation of a cancer cell. All right, one last itty-bitty small topic here before we're done. I want to talk briefly about how prokaryotes divide. Up to this point, we've talked mostly about eukaryotes, so plants, animals, and fungi, protists. But we can't leave out the bacteria and the archaea, the prokaryotes. And they don't really mess around with all their cell divisions. They just get right to the point. They undergo not mitosis, but they undergo something which is called binary fission. And it looks something like this. Basically, bacteria only have one chromosome, just one loop of DNA they don't have like a compacted chromosome like we have. It's just kind of like a, I mean, it can be compacted, but it's not like a discrete several series of chromosomes. It's just one big mass of DNA in a circular form. Now, what can happen is that at some point this ring, it gets a little split in it, and then there's these enzymes that go along the middle in between the two sides of DNA, and they replicate it. You don't have to know about all that process. That's more microbiology stuff. But the big idea is that from one big chromosome, you form two chromosomes. And then basically the cell just splits. So it's super fast. It doesn't involve all these chromosomes condensing and these mitotic spindles being formed and chromosomes lining up at the metaphase plate and all of this stuff. It just, chromosome divides, or the chromosome is replicated, and the cell divides. It's really simple. And because of that, it can happen super fast. So this is sped up, but it gives you kind of the idea of just how fast some of these bacteria might be able to grow. So you can see in no time at all, there's a lot of bacteria that can kind of come up in a hurry. Now this is super sped up, so don't be too scared right now. It's not like the universe is going to end, but it is pretty fast. Now, keep this in mind. This is a true statement. Under ideal conditions, some bacteria can reproduce every 20 minutes. Okay, so that means 1 goes from 2, 2 goes to 4, 4 goes to 8, and 8 goes to 16, 16 goes to 32, 32 goes to 64, and then from 64 we go to yada, 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 yada. Well, if this is happening every 20 minutes, assuming that there's a 24-hour day, and that's the rate that's held every 20 minutes, one bacteria in 24 hours will turn into this many. 2 gajibillion zillion of them. That is a huge number. And that's powerful. And we can sometimes be overcome by that power, different bacterial infections. If just one bacteria gets in and finds a little nook or cranny in your body that's away from the immune system, it can start dividing. In a hurry, one bacteria can turn into an army. But similarly, we use this to our advantage as well. For example, the hormone insulin that people with diabetes would take to lower their blood sugar, that is mass produced by bacteria. Basically, huge vats of bacteria are grown up. They each have a gene that produces insulin inside their genome, and because of that, they produce the insulin hormone. And you can take one bacteria and grow it up into a whole legion of bacteria that are produced like little, basically medicine-producing factories. So that's kind of the either or side of the picture. It's kind of the good and the bad, the good and the evil of prokaryotes. But in either case, that is binary fission. It's different than mitosis. One last thing that I probably should have mentioned at the very beginning. Not all cells are going to divide at the same speed or the same frequency. For example, skin cells on your skin are dividing like once every day about, whereas like some cells like in your liver or in your brain may or may not ever divide. Like a lot of neurons in your brain never divide throughout your entire life. And so that can kind of vary. The amount of time that a cell spends in that G0 phase, just kind of doing its thing, varies. That's all for now.